Translation questions on the MCAT can feel overwhelming. Ribosomes, tRNA, codons. It's easy to lose the big picture. But what if you could see protein synthesis as a simple, step-by-step process instead of a wall of details? In this video, we'll break down exactly how mRNA, tRNA, and ribosomes work together to build proteins, so you can confidently handle both passage-based and discrete questions. Let's turn translation into one of your strongest topics. Translation is the process by which ribosomes read mRNA to synthesize polypeptides, using tRNA as an adapter molecule. This process occurs in the cytoplasm and represents the second major step of gene expression following transcription. The ribosome moves along the mRNA in the 5' to 3' direction, decoding codon's 3 nucleotide sequences into specific amino acids. Transfer RNA, or tRNA, acts as the adapter that links nucleotide language to amino acid language. Each tRNA carries a specific amino acid and has an anticodon that base pairs with the mRNA codon. A helpful way to remember this is codon on mRNA, anticodon on tRNA. The ribosome itself has 3 key sites: the A site for amino-acyl tRNA entry, the P site for peptide bond formation, and the E site for exit. On the MCAT, you are often asked to identify the directionality of translation or the role of each molecule. So remember, mRNA provides the code, tRNA brings amino acids, and the ribosome catalyzes peptide bond formation. This connects closely to transcription and the genetic code, especially understanding redundancy and start-stop signals. Translation occurs in 3 stages: initiation, elongation, and termination, each involving specific molecular interactions. During initiation, the small ribosomal subunit binds to the mRNA near the start codon, typically AUG, which codes for methionine. The initiator, tRNA, carrying methionine pairs with this codon, and then the large ribosomal subunit joins to form the complete ribosome. Elongation is the repeating cycle where new amino acids are added. A charged tRNA enters the A site. A peptide bond forms between the growing chain in the P site and the new amino acid. And the ribosome translocates, so the tRNAs shift positions. Termination occurs when a stop codon, UAA, UAG, or UGA, enters the A site. Instead of a tRNA, a release factor binds, causing the completed polypeptide to be released. A mnemonic for stop codons is "You are annoying, you go away, you are gone." On the MCAT, questions often test your ability to identify what happens if a mutation affects one of these stages, or if a stop codon appears prematurely, linking to concepts like nonsense mutations. The genetic code is degenerate and universal, meaning multiple codons can encode the same amino acid while being conserved across species. Degeneracy refers to the redundancy in the code, for example. Several codons can specify leucine, while universality means that most organisms use the same codon assignments. This redundancy often protects against mutations. A change in the third base of a codon may not alter the amino acid, known as a silent mutation. The wobble position, typically the third nucleotide in the codon, allows flexible base pairing with the tRNA anticodon, explaining this redundancy. A useful tip is to think of the third base as the forgiving position. On the MCAT, you may be asked to predict the effect of point mutations on protein structure, distinguishing between silent, missense, and nonsense mutations. This concept ties into evolution and molecular genetics, highlighting how conserved processes like translation support life across diverse organisms. tRNA charging and codon-anticodon pairing ensure accuracy in amino acid incorporation during translation. Before translation even begins, Each tRNA must be charged with its correct amino acid by an enzyme called amino-acyl tRNA synthetase. This enzyme is highly specific, recognizing both the tRNA and its corresponding amino acid, and uses ATP to attach them. This step is crucial because the ribosome does not check whether the amino acid matches the codon, it only ensures proper codon-anticodon pairing. Therefore, accuracy depends heavily on correct tRNA charging. Once charged, the tRNA enters the ribosome and pairs its anticodon with the complementary mRNA codon through hydrogen bonding. On the MCAT, a common trap is assuming the ribosome verifies amino acids directly. Instead, remember that fidelity comes from the synthetase enzymes. This topic also connects to enzyme specificity and energy usage, as ATP hydrolysis drives the charging process. Ribosomes catalyze peptide bond formation and can be free in the cytosol or bound to the rough endoplasmic reticulum, influencing protein destination. The ribosome is a ribozyme, meaning its catalytic activity is carried out by rRNA rather than protein, specifically in forming peptide bonds between amino acids. Ribosomes can exist either free in the cytoplasm or attached to the rough ER. Free ribosomes typically synthesize proteins that function within the cytosol, while bound ribosomes produce proteins destined for secretion, insertion into membranes, or transport to organelles. A signal peptide on the growing polypeptide directs the ribosome to the rough ER. A helpful way to remember this is free stays inside, bound goes outside. On the MCAT, you may be asked to predict where a protein will localize based on its synthesis location or signal sequence. This concept connects to cell biology topics like the endomembrane system and protein trafficking.